Hi, this is Mr. Wartsky again, and this is the second screencast in the prokaryotic series. We ended with this slide last time talking about the difference between asexual and sexual reproduction, and I said that the prokaryotes asexually can divide or reproduce through binary fission. And here's a picture of binary fission. It'd be a great picture to draw out, and it does rhyme with division, fission and division. That's how I remember instead of fusion, which you join, fission, which you divide. So we have the DNA copied, forms two nucleoids. They go each into a different cell, and that is binary fission. So sexual reproduction, though, is a little different. We need to remove all basic concepts of what we consider sexual reproduction in our minds. So there are three ways, yes, there are three ways that prokaryotes reproduce sexually. Three ways. Now, this you needs to have something known as the F plasmid, which is the fertility plasmid. If a cell prokaryote has the fertility plasmid, it is called F plus. If it does not have the fertility plasmid, it is known as F minus. Now, there are ways to transfer the fertility plasmid or the F plasmid from an F plus to an F minus, and that is through a pilus. So the plasmid will, a copy of the plasmid will move through the F pilus that connects the F plus and the F minus into the F minus. And when the F minus gets a fertility or F plasmid, it becomes F plus. It is not yet sex. Nope, because we have not mixed up or combined the DNA from the two sources. We have a plasmid that is surviving on its own, individual of the nucleoid. Now, sometimes there is a HFR cell that's formed from an F plus cell. And make sure you get all this down. So HFR stands for high frequency recombination. And how do you get an HFR is when the F plasmid joins with the nucleoid. Write that down. The F plasmid joins with the nucleoid, and then it becomes an HFR cell. Now, the HFR, not sex yet, okay, the HFR forms an F pilus. Instead of just having the F plasmid go through the F pilus, instead the whole nucleoid will enter the new prokaryote, the F minus prokaryote from the HFR cell. Now, the nucleoid from the HFR cell will join with the nucleoid of the F minus cell. And when we have the joining of the two nucleoids, that is sex. So if you need to listen to that again, you might want to. Now here is a picture of conjugation. We have the F plus plasmid up here. It will enter, and then we have just an F plus. This will become an F plus. This is now an F plus, and we're two F pluses. The HFR, this F plasmid joins with this nucleoid, and the HFR cell forms an F plasmid, or F pilus, and donates the whole nucleoid, and then the nucleoids combine, and when the nucleoids combine, that is sex. Right there. We have the HFR cell. The red area here is the F plasmid. It goes through the pilus, and then it combines. Right there. Sex. Transformation occurs when a prok dies. DNA's out there. A new prok comes along, absorbs the DNA of the dead prok. Okay. Now there's two types of transformation. There's natural, and then there's artificial. And the natural, the bacteria absorbs the DNA from a dead prokaryote. Naturally happens in hospitals a lot. That's how you get the antibiotic resistance. And then artificial, we can actually place plasmids with the genes that we want on the plasmid into a prokaryote. And we're going to do that later on in the year in the recombinant DNA section because we're going to review artificial transformation. Transduction, the virus carries prok DNA to a new prokaryote, injects the prokaryotic DNA into the new prokaryote and the DNA mixes, and you have sex. So the three ways that you can have sex in a prokaryote is conjugation, transformation, and transduction. Now on this slide, we have bacterial transformation. We're going to do this in the class. Don't worry about it. Just make sure you know that DNA is absorbed from a dead prok or with the insertion of a plasmid. Here's transduction, in which a bacteriophage is the only type of bacteria that attacks virus that attacks bacteria will insert prokaryotic DNA instead of viral DNA into a prokaryote. So in our discussion of prokaryotes, we're going to talk about metabolic diversity, how the prokaryotes produce energy in order to survive. And as we'll find out later, that's your whole goal in life, is to produce energy, because the energy will keep you alive. If you don't produce enough energy, you will die. So we need electrons to produce energy. The shorthand for energy is E. So heterotrophs, we are dependent upon outside sources for organic molecules as well as electrons to produce our energy. Autotrophs, they produce their own organic molecules from inorganic carbon to produce the electrons for energy. Phototrophs use the energy from the sun to produce organic molecules from inorganic carbon, and chemotrophs use the energy from chemicals to produce organic molecules from inorganic carbon. So, like for example, photoautotrophs like plants use carbon dioxide, which is an inorganic carbon source, and the energy from the sun to produce an organic molecule, glucose, which is then used for fuel in the energy production. I just said that before. Photoautotrophs, we combine these terms, uses light to produce organic molecules from inorganic carbon. The organic molecule is then used as a source of electrons to produce energy. That's all glucose is. It's a source of electrons so we can produce energy. Photoheterotrophs, they use light to produce organic molecules but need an organic source of carbon. So they can't use CO2 as their source of carbon. They have to use something else, some other carbohydrate, some other protein, or some other fat to produce the electrons necessary or to produce the organic molecules so that can produce the electrons necessary for energy production. Now, that only happens in prokaryotes, not really discussed a lot. We're not going to really talk about that a lot either. Chemoautotrophs. Now, these are those bacteria that live near the deep sea thermal vents. They use the energy from chemicals, usually from hydrogen sulfide. Right? The electrons, they grab the electrons from hydrogen sulfide and they use those electrons to produce organic molecules from inorganic carbon. The organic molecules are used as the energy electron source to produce energy. Again, those are only found in prokaryotes. So instead of the sunlight, hydrogen sulfide is used. Now, chemoheterotrophs, we need to consume our organic molecules for the electrons that break them down. So we're going to use glucose, right? So we take in glucose, we break glucose down. The glucose provides us with the electrons necessary to produce energy. And we'll talk about that when we talk about energy production. So oxygen requirements in prokaryotes. We can have obligate aerobes, which need oxygen. That's what obligate aerobes mean. We can have obligate anaerobes, which can only survive in oxygen-free environments, and facultative anaerobes, which can survive in the presence of oxygen, but doesn't need oxygen to survive. So it can survive in both oxygen and oxygen-free environments. So from prokaryotes, which is a large category, there are two types of prokaryotes, eubacteria and archaebacteria. Eu is true bacteria, archaebacteria is ancient bacteria. Everything that we have talked about has been eubacteria. So we're going to talk a little bit about archaebacteria. So archaebacteria, they don't have peptidoglycan in the cell walls. So that's different than in eubacteria. The cell membrane has a unique lipid composition, and they have different types or different structures in the ribosomes. All the ribosomes have a slightly different structure, which again is going to be important in a little bit. So here is a comparison chart between eubacteria, archaebacteria, and eukarya. Now eukarya are eukaryotes. We are eukaryotes. So I want you to get this table down, put it on pause, and this is the last thing for our prokaryotic screencasts. So if you need to watch them again, please do so. Good night.